and we just went over the lytic cycle of bacteriophage replication. And I challenge you to go back through that, make sure you understood it before we layered on the next piece. So we went back through here. This is the lytic cycle. And again, lots of different bacteriophage use this lytic cycle. This particular one is a T4 bacteriophage that invades E. coli. And this is where all the studies were done to understand this. So this is one type of bacteriophage called the T4. There are others. And this is the type of replication cycle it goes through. And it's called a lytic cycle. Now, I've mentioned that if a bacteriophage enters, that cell is going to die. Okay, it is going to die. Once a virus enters that cell, it is marked for death. The lytic cycle is a very quick method of death. Gets in there, right? 15, 20 minutes, 20 minutes, 30 minutes or something. It gets in there and then it is going to release by lysis. There is another type of replication method done by other types of bacteriophage called the lysogenic replication cycle. And this one has a lot of similarities and there's some differences. So let's learn about the lysogenic. The lysogenic cycle, again, once a virus enters, a variant enters, that cell will eventually die. But in the lysogenic cycle, it actually slow plays it. It allows the bacteria cell to live and even replicate for a time period before it dies. And we'll see how that works. So lysogenic, think of that as a slow cycle, a slow death. Lytic, lysis, very quick death. So let's see the differences in the lysogenic and the lytic cycle. All right, a lot of similarities. Attachment, same thing, exact same thing. So you get attachment. Entry, same thing. That bacteriophage enters, the nucleic acid only enters, you get a ghost phage left behind, same thing. This is where the difference comes into. If a bacteriophage undergoes the lysogenic replication method, instead of immediately going into synthesis, assembly, followed by release, it enters a new stage called lysogeny. And in this stage, the bacteriophage is integrated into the bacterial DNA chromosome. So the viral DNA gets integrated into the host bacterial DNA. And then actually the virus lives for a while and can even replicate. So if I even go back to my drawing here, the viral nucleic acid has entered. In my bacteria cell is my bacterial chromosome doing that. What happens to the nucleic acid after attachment and entry is the viral nucleic acid gets integrated into the host chromosome. So this host chromosome now contains some genes from this viral nucleic acid. So instead of the virus going ahead and making enzymes that are going to cause lysis, it just integrates. And then the virus doesn't do anything. The bacteria cell gets to live. It can even replicate. So if this cell replicated, it can go through binary fission, right? And make two cells. And each one of those cells is going to have a chromosome in it, and each one of those chromosomes is going to have the viral nucleic acid in there. So if you can see this, I got my cell, went from one cells to two cells, chromosome replication, right? We know how that works. It also replicated the viral nucleic acid. You can't really see the red. Let me make that a little bigger. It also replicated the viral nucleic acid when the bacterial chromosome got replicated as well. So it can go on for that. And honestly, it can go on like that for a long time. Virus, what's the point of it? Well, viral nucleic acid is getting replicated, which that's kind of the goal. All organisms want to pass on their nucleic acid to their progeny. That reproduction, replication, that's the goal. So the goal is still being made. So it's fine with that. So any of these progeny is going to contain the viral nucleic acid. Now there is a vocabulary term for that. When the virus, remember we've defined virus, we've defined virion. A virion is a infectious virus outside of the host. When a virus DNA or a nucleic acid It has to be DNA to integrate. When the DNA, viral DNA, is integrated into a host chromosome, we now refer to that as a prophage. So this viral nucleic acid, remember, it doesn't have a capsid or anything, it's just nucleic acid. It's integrated, it's protected by the cell. We call this a prophage. So it's viral nucleic acid integrated into viral DNA, integrated into the host cell DNA. Now this only works if this bacteriophage has a DNA nucleic acid, okay? Because we can't integrate RNA into the DNA double helix. Okay, so back to lysogeny, the lysogenic cycle. We have attachment, we have entry, but this time the viral DNA is integrated in the bacterial DNA, and then the cell goes through binary fission, and the viral DNA gets copied along with it. So this is new. Now, I said it can happen for, it can stay that way for a long time, but at some point, there may be an environmental stimulus that's going to cause the prophage to excise itself, come out of the bacterial DNA. And then when that happens, it goes right back into synthesis, assembly, and release. So it goes right back into making this happen. It goes right back into those lytic phases of the cycle. Why would this happen? Okay, well, say that bacteria cell has been living just fine and going along well, and here you come with a chemical and you're spraying Lysol all over it. That bacteria cell is going to die. That chemical will trigger induction. Induction is when the prophage excises out of that bacteria cell, goes through synthesis, assembly, and release. It's a survival mechanism for the virus. The virus has been replicating fine within the bacteria cell, but if that bacteria is going to die, that virus needs an exit strategy. That's what induction is. Some sort of environmental stimulus that's going to cause the cell to die or be in danger, and that virus is going to get out. An induction. What else can do that? You know, the same things that mutate DNA, chemicals like your Lysol, X-rays, UV radiation, those same types of things that damage nucleic acid will cause induction to come out. So those types of damage. If we're talking about well, we'll talk about that, and we'll talk about that when we get to eukaryotic things. So anything in the environment that's going to damage or put that cell in danger is going to be an induction and cause that to come out. All right, so what happens after that? After that, it comes out, synthesis, assembly, and release by lysis. Okay, so let's look at the pictures, and I want you to go through the picture and go through all the steps and see if you can follow the lysogenic cycle. So I'm going to let you do that. All right, so how'd you do? Hopefully you got through the steps. Attachment, the same thing. This lambdaphage is an example of a bacteriophage that undergoes this lysogenic method. We got attachment, we got entry, and then instead of immediately going into synthesis, assembly, and release, it went into lysogeny. The viral DNA integrated into the chromosome, and now we call that viral DNA a prophage. Life continues, replication occurs, binary fission, until some sort of environmental trigger triggers that prophage to come out, excise, and then it goes right into synthesis. It makes that nucleic acid, which degrades the bacterial chromosome. It makes capsomeres, it makes glycoproteins, it copies the viral nucleic acid. We put them all together, and then we get release. All right, so when I said when a virus enters a cell, it's eventually going to be a goner, it may not be immediately, and it could be that cell plus all of its progeny. But if a virus is present in that cell, it's eventually going to die. There's no way to protect itself from that virus, okay? All right, so this is where we would stop, and you would go to your study groups in class, and explain that. So definitely take the chance to do that with your study groups and talk out these two things. Again, these are processes. It just helps to talk out. Here's a phenomenon that we see called lysogenized conversion. So you remember the lysogeny replication method, that you have that viral nucleic acid that is integrated into the bacterial chromosome for that. Again, remember back here, I've got my viral nucleic acid integrated into that bacteria. So that can make a difference in this bacteria. It's got something new in it. It may have interrupted some other things. It can make a difference in the behavior of this bacteria, and we refer to that as lysogenized conversion. This bacteria, because it has undergone lysogeny and has this prophage, it's going to be converted in some way. It's going to have some different characteristics. So let me give you a couple examples of lysogenized conversion. So one example, there's a bacteria called Streptococcus pyogenes. If we get invaded by Streptococcus pyogenes, this is a bacteria that can cause strep throat. We get it, we get strep throat. How do we fight strep throat? You know, most of you have had some personal experience with this. You go to the doctor and you get an antibiotic. So Streptococcus pyogenes gets invaded, causes strep throat. But Streptococcus pyogenes can be used as a host for a specific virus or a specific bacteriophage. This particular bacteriophage invades the bacteria Streptococcus pyogenes and undergoes lysogeny. So it integrates and becomes a prophage in the Streptococcus pyogenes. Now, if we get invaded by this lysogenized Streptococcus pyogenes, so a bacteria that contains this particular bacteriophage, we don't get the disease strep throat. It actually changes how Streptococcus pyogenes infects us and the damage it can cause, and actually results in a different disease called scarlet fever. Now, you may not remember much about scarlet fever because we don't see it very much anymore, because it used to be a disease that infected, before antibiotics, so before 1940s, people would die from scarlet fever. They would get it, causes high fever, a lot of similarities to strep throat, but even some additional ones, causes a big rash all over. And again, the mortality rate was much higher. People would die from scarlet fever, or they could have other severe side effects like blindness and other things that could come from that. Nowadays, we still get the disease scarlet fever. It still pops up, but we have antibiotics. So we get rid of it very quickly before that bacterial disease can cause those damaging side effects. But what's going on now is that the same bacteria with a bacteriophage causes a completely different disease. So that's an example of lysogenized conversion, how that bacteria has different properties and can do different things because of the presence of a bacteriophage. So that's one example. There's another one. Your vaccine DTaP or Tdap, depending on whether you're a baby or an adult. It stands for diphtheria, tetanus, and pertussis. So vaccine that protects us against those diseases. Well, diphtheria used to be a bad disease. Again, it's caused by bacteria, so now we just treat it with antibiotics. But the bacteria that causes this is called Corynebacterium diphtheriae. If it is just the bacteria and no bacteriophage involved with it, it actually doesn't even cause a disease. It doesn't cause a disease at all. It's only if Corynebacterium diphtheriae becomes lysogenized, has a prophage, a bacteriophage integrated into that it even causes the disease. So lysogenized conversion is a way where it changes the bacteria and leads into a different outcome for that, because now the bacteria has some different characteristics. Okay, lytic and lysogenic phase. Go back through those. Those are big ones. Talk them out and get them down, and then I'll see you in the next video.